Hi, folks. It is I, the floating head. And I'm here today to talk to you in research methods about single factor experiments. So, we're going to spend a little bit of time talking about experiments because, by and large, they are probably one of the most powerful ways, at least in psychology and other social science fields, to actually learn about the world and draw cause effect relationships. So, we're going to talk about a few different sample studies, and then I would also like you to think about our own replication study, which will go on in the fall, that we will put together. And I'd like you to think about the ways that our replication study is technically an experiment. I want you to think about why, and I want you to think about what kind of experiment it is. So here's what we're going to cover. So we're going to talk about a few different examples. Oh, hi. Hi, research methods. Your TA Earl was on my lap, but as soon as I put the camera towards him, he was like, "No, not having that." Reactivity. So we're going to talk about different examples of single factor experiments. We're going to talk about different types of experimental variables. So we're going to go over the difference between an independent variable and a dependent variable. And we'll talk about the different ways that experiments actually support cause-effect inferences. As I mentioned, experiments are really the only type of research study that we can do that we can actually draw causal conclusions from the study. So we're going to talk about the different reasons why that is. We'll talk about between and within subject designs, and then we'll talk about those 4 different types of validity, like we did with correlations, and we're going to talk about how each of them plays a role in experiments. So we're going to start with a couple of different examples of what are called single factor experiments. And I'll talk a little bit as we go through each of these studies, what makes them what we call a single factor experiment. So first, I know it's a little bit harder to kind of do this now, but I want you to think about when you were taking in-person classes with me or with other professors before spring break. Some of you are very, very fond of using laptops. Some of you are very, very fond of taking longhand notes using a pen or a pencil and paper. I am definitely of the camp that I like to take notes in longhand just because my pen or my pencil and my paper do not run out of batteries, and at least when I've taken notes on a laptop or taken minutes on a laptop, if I don't have a charger, I inevitably end up running out of battery power. Now, I bring up this study because here's something that's actually kind of serendipitous and kind of lucky. So I adopted this textbook. This is the first time I've used the 3rd edition of Morling for Sci 310. What's interesting is that this exact same study is talked about in your textbook that we're using in the fall, Introduction to the New Statistics. So they're going to talk about the data a little bit differently, but I thought this was really cool that the same study gets brought up in both of your textbooks. So don't forget about this study because it will pop back up in the fall. So this was a study by Mueller and Oppenheimer in 2014. And what they basically had people do, they were either assigned to take notes using a laptop, so they were in a classroom. Half of the students were randomly assigned to take notes on a laptop. The remaining students in the class were assigned, or in the experiment, were assigned to take notes using longhand. So either pen or pencil with paper and taking notes. And so obviously you can think about some of the major benefits that a laptop will give you when you take notes. Because you tend to type a little bit faster than you write, you've got a better chance of getting everything that the instructor says like verbatim. With longhand, you obviously can't take down everything verbatim, so you have to make choices about what's important and what really isn't. And so both of these methods have their advantages and they have their disadvantages. But what's really critical is which of these is more conducive to learning. So they had people in these groups, like I said, half were assigned to a laptop condition and the other half were assigned to a longhand condition. And they basically had to listen to a bunch of TED Talks. Now, everybody got the exact same materials. And they then had a final test. So there was a distraction periods. There was a period of, I think, about 30 minutes where they were engaged in some other tasks. So they weren't thinking about the material at that time. So it was kind of a test of long-term retention. And then they asked the participants either questions that were factual in nature or conceptual in nature and required a deeper understanding, probably greater involvement of application. So the data you can actually see over here. I will mention that these groups were randomly assigned. This is going to be very important when we talk about ruling out alternative explanations for findings. But you can actually see from the data here. So this has been done in a standardized format. Really, I think what's important to note here is the magnitude between the two groups. So notice that there's not really a lot of difference between the two groups on factual material. Now, this doesn't mean they were equally bad on facts. We have standardized these scores. 0 means that it's in relation to the mean or the standard deviation. So you can see that there's really no difference between the groups in terms of factual content. What's interesting and probably pretty important is the difference in conceptual. So a positive standardized score means that you're doing really well. A negative score means that you're doing much, much worse. So you can see that the longhand group actually performed much better on the conceptual questions than students that were taking notes with a laptop. So the data, and they did actually do a few follow-up experiments that found the exact same thing. So what this tells us is that even though there's no difference between groups for factual content, if you want to have a really good long-term understanding of these underlying concepts or theoretical understandings or applications, so if you have to do that deeper kind of thinking, it's better for you to actually take notes with longhand than using a laptop. I don't know that this will necessarily stop those of you that do laptop notes from using your laptop, but maybe it'll make you think about it a little bit. So here's another example. Do you like pasta? I think most people do. I've yet to meet a person who really doesn't like pasta. This comes from a study by Van Cleef and colleagues in 2012. So this is a case where they were curious as to whether the size of the serving dish that you serve yourself from, would that affect how much you ate? So one of the things that often tends to come up when we talk about how much people eat, and especially here in the United States, where we have a tendency to eat a little bit too much, and there's also data that shows that portion sizes, particularly at restaurants, has increased over time. Does the size of the serving plate actually affect how much you eat? So participants were basically put in these conditions where they had to serve themselves pasta, not past, from a communal bowl. And these participants were either randomly assigned to a medium-sized bowl that they had to serve themselves from or a large bowl. And so these researchers looked at how much pasta they served themselves, and then they weighed the bowls with leftover pasta to basically make a determination of how much people had eaten. And generally what we find is that when you have to serve yourself from a larger bowl, you tend to take more. So in this case, the group in the large bowl took almost 200 grams worth more of pasta, basically taking 140 kilocalories, or just plain calories with a capital C, more than the medium-sized group did. So if you're trying to work on eating a little less, this data suggests that you might want to use a smaller bowl. And because it was an experiment, we can be pretty certain that this is a cause-effect relationship. Now, why do I like experiments? Experiments, by and large, are the gold standard of research. So, one of the things that makes experiments really cool is how much control you have over the data that you want and the data that you collect. So, it allows us to exercise experimental control. We get to choose what we're going to manipulate. So, when we call these single factor experiments, by the way, we are only manipulating one factor or one variable. So in the Mueller and Oppenheimer study, our independent variable, our single factor, is the manner in which you have to take notes. For our pasta study, it's the size of the bowl. Is it medium or large? And that's the only thing we're manipulating. That's why we call it a single factor experiment. So we get to choose what we manipulate. We also get to choose what dependent variables we want to use and how we want to measure them. And we are going to do as much experimental control of all other aspects of the environment that we can, so we don't get any confounding variables that could potentially dampen or cloud that cause-effect relationship that we want. So, for example, in the note-taking study, everybody listens to the same TED Talk. They're all in the same room. In the pasta study, everybody's getting the same set of experimenters and they're all getting the same type of pasta. So we want to make sure as much as possible that aside from our independent variable, that we're holding all other variables constant. And we'll talk about some different ways that you can do that in a little bit. Now, additionally, and why I love experiments, is it allows us to draw cause-effect relationships or causal inferences. So, if you are trying to see if something in the world is actually real and if you want to see if an effect is real. Ideally, if this is something that is real out in the world, as you vary your independent variable, you should see some change in your dependent variable. Your independent variable is always going to be manipulated before you get those results, and control rules out confounds. So we either need to keep those extraneous factors constant across participants, or we need to balance whatever we can't hold constant. So these are the three criteria for causality. I will go back and talk about this a little bit more in a bit, but experiments meet all of the three criteria for causality if you are designing your research well. And that's why experiments are pretty great. So let's now spend some time talking about these different experimental variables. So experiments have both measured and manipulated variables. And a manipulated variable occurs when the researchers will assign a participant to a level or a particular condition of the variable. So the manner of taking notes in the Mueller and Oppenheimer study, there are two different levels. You are either taking notes with a laptop or you're taking notes longhand. In the pasta study by Van Cleef and colleagues, you are either serving yourself pasta from a medium-sized bowl or you're serving yourself pasta from a large bowl. So those are what we are directly manipulating. We are assigning some participants to some conditions and assigning other participants to other conditions. We also have a measured variable. We're going to record what happens in terms of changes in attitude or changes in emotion or changes in behavior. And this can include things like self-report. This can include various behavioral observations, things like accuracy, reaction time, behavior that's being coded. So, for example, in the Oppenheimer study, the notes and the answers to those factual and conceptual questions were scored for correctness by a group of readers. So those data were a form of behavioral observation. We can look at physiological measures like if we were doing fMRI or EEG. So when we talk about our manipulated variable and our measured variable, we tend to use very specific terms. So our independent variable, or our IV, is what we are manipulating. So that manipulated variable is our independent variable. When we talk about different conditions, we are talking about the levels of the independent variable. Now, both of the studies that we've talked about so far, the independent variable has two levels. You are either getting a laptop to take notes or you're getting a pen and paper. Our dependent variable is functioning as our measured variable. It is the outcome that we are looking for. So ideally, if we have an effect and that effect is true, changes in the independent variable, so being assigned to a different condition, will lead to differences in our dependent variable. Now, I did mention the importance of controlling extraneous variables, and these are what are known as control variables. These are what I am trying to hold constant across all of my participants, and I'm making sure as much as possible that my participants are experiencing largely the exact same environment, save for that one independent variable. Now, I will mention that calling it a control variable is a bit of a misnomer. We're not actually varying these things, so they're not technically variable. But we do have control variables. So, for example, regardless of what note-taking group you're in, everybody gets the same TED Talk. Regardless of what bowl condition you're put into, everybody gets the same type of pasta. So we need to keep those controlled because we want to make sure that it's only the independent variable that's causing a change in our dependent variable. So why do experiments allow us to make these causal claims? Well, let's go back to our three criteria for causal claims. So first of all, we have the principle of covariance. As one variable varies, you should see the other variable vary. Now, when we were talking about this with correlations, that's the same sort of idea. So as one variable changes, the other variable should change. We have our criterion for temporal precedence. And this is where correlations and correlational research really starts to fall a little bit short. With temporal precedence, the change in one variable has to precede the change in another variable. And remember that because of bidirectionality with correlation coefficients, we can't really figure out what variable came first. With experiments, because we're manipulating the independent variable first and then measuring the dependent variable, we always know which variable preceded the other. And then finally, we have the criterion of internal validity. We need to rule out any other possible explanations for how we got the results the way we did. So we want to make sure that there aren't any other extraneous variables or confounding variables that could cloud that cause-effect relationship. Again, with correlations, this is another area where we fall short. So for example, if I have a correlation between violent television, so watching violent television and becoming more aggressive or more violent, there may be a third variable, such as something like parental involvement, that might explain the behavior of both of those variables. But because correlations can't rule that out, we can't really make a causal claim. If I'm using a well-planned study and I'm doing enough experimental control, I should be able to rule out any possible alternative explanations for why my experimental results shook out the way they did. So, why do experiments support those causal claims? Well, experiments establish covariance. Ideally, if I get an effect, a change in my independent variable should cause a change in my dependent variable. Additionally, experiments establish that temporal precedence. My independent variable changes before my dependent variable does. And as I said, well-designed and well-controlled experiments rule out any possible alternative explanations for the data. So the only thing that could have caused that change in the dependent variable is my manipulations in my independent variable. So let's spend a little bit more time talking about each of these in more detail. So one of the things that we've kind of been talking about, and I think your book did a really good job talking about at the very beginning of the semester, is that it's really, really important to think about your comparison groups or comparison conditions. So we talked a little bit about bloodletting with leeches as an ancient treatment, and a lot of the evidence that researchers and early physicians had for leeches being a good bloodletting treatment is that, well, using my anecdotal evidence, whenever I give somebody leeches, they always seem to recover. That's a terrible comparison group because you're comparing it to people that haven't had leeches and ended up not recovering. But there are 4 possible outcomes there. We can have people who had leeches and recovered. We can have people who didn't have leeches and they ended up dying. We have people who got leeches and died, and we have people who were not given leeches and recovered. So we have to think about comparison groups. Your book talks about an instance where researchers looked at pictures of people hugging dogs and reported finding that a lot of the dogs in the photographs showed evidence of signs of stress. And they used that as evidence for dogs not liking to be hugged. And that's fine, but they didn't look at instances where dogs were not being hugged and whether or not they were showing signs of stress. So you need a good comparison group and independent variables. By setting up these different levels of comparison actually give us a group to compare to or a comparison condition. So so we have that covariance. We actually get a comparison group that varies. Additionally, we need to think about the results. We need to see a change between our groups in that dependent variable. Otherwise, we don't have an effect. Can you imagine in the laptop versus longhand notes study, if there were no differences between the groups, not only on factual or conceptual? So in this case, there's no covariance in the DV, and that means there's no effect. So if we want to get an effect, we not only have to have covariance in our independent variable to establish that comparison group, we also have to have covariance and variability in our dependent variable if we want to see an effect. Additionally, we can use control groups as another basis of comparison. So under certain cases, we can have a control group where there is no treatment. Now, we have to be careful with this because sometimes there's not necessarily a very good control group that we can have. So with the note-taking group, we could have a group that didn't take any notes at all, but that seems like that would be a really bad strategy for a college student. So in that case, we don't really have a control group. So sometimes you need one, sometimes you really don't. We can have a treatment group, which are basically different groups that are being exposed to levels of the IV. And if we're looking at research utilizing particular interventions like pharmaceutical interventions or drugs, we usually want to have a placebo group because we want to see we want to be able to separate out the biological effects of a drug or a treatment from the psychological benefits and beliefs of being treated at all. So a placebo group can help us determine, does the drug actually work because it's affecting biology, or is it merely working because people want it to work? I'm going to go ahead and stop here. We'll talk some more about experiments very soon.